Hi, this is Miss Linton. This is my period 1 again. Say hi.
Hi.
All right, and we're going to talk about permeability. And so permeability of the plasma membrane, first thing you want to know is only certain molecules can pass through. You shall not pass. Sorry, little Lord of the Rings reference. Okay, only certain molecules can pass through. So you can see right here, phospholipid bilayer. You're focused. Phospholipid bilayer, what are these things right here? Proteins. So the things that cannot get across a cell membrane is if they are large or charged. Okay, so who can't get through? If you are large or charged. See how I'm doing like plus, yeah, good. Large or charged. So if you are, tell me again, large or charged, okay, you have options. Okay, your options are to use a channel or a carrier. Okay, or a third way would be whole membrane, where you do endocytosis or exocytosis. Okay, got that? Now, if we look at this diagram right here, you can see charged molecules turned away. Water can get through, but water is very small, even though it is polar. Okay, it can get through. Non-charged molecules, fats dissolve into fat, so they can go right through, because this is the part that's going to turn you away. Okay, this hydrophobic region, that's the one that kind of trumps them all. Okay, and so, so I'm going to build a wall, and I'm going to make these proteins pay for it. Okay, so anyway, so all right, so these non-charged molecules, they can pass through freely. These large macromolecules, they can't get through. This creates a barrier, and then I can control their movement based on whether I'm doing exocytosis or whether or not I'm allowing them to use a carrier. Okay, so that's how you can control the movement. So on your notes, in general, what can pass freely through a phospholipid bilayer? Water, gases, small, non-charged, usually non-polar molecules. Water, gases, small, non-charged, usually non-polar molecules, and hydrocarbons. What can't pass through without help? If you're large or charged. Youngest bio buddy, could you explain this to him real quick? Do a little summary. What can pass, what can't pass? Go. Yes, baby.
How does that charge molecule on the top right hand?
Oh, could have been a carrier. Yeah. All right. So I'm going a little bit out of order because I don't know why I have this next slide, so I'm going to skip here in case you're in the thing. There are two types of transport, passive and active. Passive, it does not require any what? Energy, whereas active transport does. So let's start by looking at some examples of passive transport. This first one is diffusion. Diffusion is just when molecules move from a higher concentration to a lower concentration. So when you have diffusion right here, these molecules are going from a higher concentration through this phospholipid bilayer to the other side. They must not be and they must not be charged, so they're able to move through. Okay, it could be oxygen, it could be carbon dioxide, it could be water. And it's just passively going from one side to another. It doesn't cost your cell anything. Here's your second one, your second option here. It's facilitated diffusion. Facilitated. Something's facilitating it. What's facilitating it in the next two? What are these things in the membrane? They're proteins. This is going between the phospholipid bilayer. This is going from one side of the membrane to the other, but it's using a what? Protein. Notice this one would be a what? Channel, and this one would be a carrier. But in this case, is it costing the carrier any energy? No, because it's going from a higher concentration to a lower concentration. These are all under the umbrella of passive transport. Higher to lower, either between the phospholipids or using a protein, either a channel or a carrier. Okay? Active transport. There are two types of active transport we'll talk about. This is only one picture. I am using a what? Carrier, and I'm going against the concentration. I'm going from a lower concentration to a higher concentration. I'm pushing the balls uphill. It takes more energy to concentrate them on that side. That is one form of active transport. Here is another. What is this? Endo and exocytosis. This is using the whole membrane. That's another form of active transport. So on your notes, where you have it, passive transport does not require what? Energy towards a lower concentration gradient. And I gave you examples right there. Jump down to number 3. Active transport requires what? Energy against the concentration gradient. Examples would be the pump, like the sodium-potassium pump, and then endo and exocytosis. Now, a form of passive transport, and this hasn't been known about for a long time, is called an aquaporin. And aquaporins, what is the problem with a water molecule? What are we running into that makes it like, hey, how's it getting across that phospholipid bilayer? It's what? It's polar. It's got it's partially charged. So you think it might have a little bit of a struggle. So how can water move so quickly? There are protein channels that are specifically set up for water movement. Okay, so here, I'm a highlighter. Meh. Research, aquaporins, I'm waiting for you. Come up. It's coming. There it is. Okay, so here you can see the structure. Now, look at that crazy stuff. What are some things you're seeing right here? Do you know what that is? It's a protein, right? And some of them are in helical structures. Do you see that? And they're trying to come up with what does that look like. Okay, and here you can see how the water molecules, it would be something like this when it passes through. Okay, here you're looking at some of the structures that they have figured out for how it might work. Okay, and then, where is that? Let me go back here and I'm going to show you the video. Minute. Where are you, notebook? There you are. Okay. Okay, so here's a little video. Oh my gosh, sorry you guys. Here's a little video showing how water might move from one side to another. And here's your key part right here. I don't know if you can see the little yellow guy in the middle. Okay, and that's because it's now we have a better understanding of why water can move so quickly, so rapidly across the cell membrane, even though it's a polar molecule. So underneath aquaporins, I have all of that for you. Whose turn is it? Whoever explained the functions of the proteins, I want you to talk about permeability of the plasma membrane. Go ahead. Yes. In that diagram, I have no idea. You do not have to keep that in your wheelhouse. I was lending it to the specificity. I'm sure we could look it up, but you will not need to keep that in your wheelhouse. 7 number on the back of your clicker.
Oh, mine just turned off.
What could you do? Welcome to my world. What do I do? I don't know. So your lab prep, is that honka honka bojamin?
It's just SAT words.
SAT words? Oh, there you go.
Oh, islands.
What's the islands good for? The basket of unlimited French fries.
Yeah.
Wait, how do you spell C?
Oh my gosh. Okay, good job, Outback Steakhouse, Panda Express, and Chili's. Why did they pick C? Could that work? Could a carrier protein be involved in diffusion? What kind of diffusion is that? Facilitated. A better answer up there is what you said. A, carrier proteins are specific. Check with your bio buddy, see if they were part of the, I was going to say anus. Now I did say it. Part of the 92%. Go ahead, ask them. Are you part of the 92?
We are.
A, you know, like A, and then I like happiness. Yeah, I should have kept that to myself. Oh, I don't know if I told you this, but it is in the, I think I did tell you this. Yes, you did, right?
I think I got it. EHS cafeteria. I think I got it. I think so. Yeah. So PHS cafeteria.
Okay, Mimi's. Mimi, show your clicker to your bio buddy. Okay, I'm done. I can't wait for you, Mimi. Oh, we dropped down to B+. Good job, BJ's, the old spaghetti factory, and Brugger's bagels. So cholesterol is found in the plasma membrane of just animal cells. I don't know why, okay, but I could guess. Let's make a hypothesis. Why do we need cholesterol for animal cells and we don't find it in plant cells? Maybe, but plants have hormones too. Yeah, nice. Plants have a cell wall to keep their act together, right? We don't have that, so cholesterol helps with the stability of that cell membrane, whether it gets too hot, kind of holds it together, too cold, doesn't allow it to what? Freeze. Okay, good job, Ruthie. All right, and let's see, I think we're moving on. We are, okay. So this is something that's going to come up in several different chapters. I'm just kind of addressing it a little bit now. This is one of your highly suggested reading and thinking, right, at the beginning. I'm sure you've already done it. Yeah. Okay, so this is underneath big idea 3, information storage, transmission, and response. We will talk about it when we talk about, let's say, hormones will be another place that we're going to talk about it. So I would like oldest bio buddy in the notes, do number 1 right now, what it says for number 1. Okay. It might have happened after, or if it's being weird, turn it off and get another one. Turn it off and turn it back on. Okay, so come back to me. So cells need to respond to their changing environment, and we've talked about how can one cell talk to another cell. It can talk to it by touching it, right? It can communicate in that way. But the only other way is it can listen, and the way you can communicate is with chemicals with cells. That's how they're going to communicate one cell to another is through chemicals. So, for instance, I mentioned earlier about the example of a what? What would this be? A receptor protein. And your anterior pituitary, you have your posterior and your anterior pituitary, and you're going to learn all the hormones of those. That's going to be in your brain, okay? And your pituitary secretes growth hormone. Now, growth hormone that it releases permeates and goes through and is in your blood supply and goes through your entire body. Yet it only impacts certain cells and tissues. Why is that? Because only those cells have the, yes, I heard it over here, whispered. What was it? Receptor, okay? Only certain cells have the receptor. So you can blanket your entire body with it, but the only responding cells are the ones that have the receptor. So that's the first part of signal transduction, is you have to have a receptor. Have we seen where this could go wrong? Yes. Because if somehow your receptor is broken, none of the rest of this can happen. It's kind of like you're going, no, 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 I can't hear you. And so nothing else will happen as a result. Then this receptor protein has to trigger another reaction, which triggers another reaction, because this receptor isn't going anywhere. It's staying put. So this, for instance, this right here could be a hormone, and that hormone could be the first messenger, which triggers a second messenger inside the cell. Or this receptor could trigger another protein that allows something else to come into the cell. And so you have a series of, like it says here, relay molecules that are passing that message along. Your end destination, it depends. It could be triggering a series of reactions that makes ATP, which is the DALA of the cell, to catalyze whatever reactions it's going to do. Or maybe the cellular response is going to activate DNA and getting that DNA turned on so you make mRNA and certain proteins that can respond to that. So there are three parts: reception, transduction, and then ultimately what your response is going to be. For instance, if I say to you, clap 3 times, that was the response, right? I sent a message. My mouth vocalized, clap 3 times, okay? When those sound waves came through your ear, your pinna directed it down your auditory canal, your tympanic membrane, your eardrum started vibrating, which vibrated your malleus, your incus, and your stapes. Your stapes was on the oval window of your cochlea. Stapes would move back and forth. Your oval window moves back and forth. All of a sudden, the fluid inside your cochlea moves back and forth. Eventually, your bacillar membrane is going to go up and down. You have hair cells embedded in a tectorial membrane. The hair cells bend, triggers an action potential, goes to your auditory nerve to your brain. There in your brain, that got, okay, so that was relay molecules, right? Here in your brain, it went, she said, clap 3 times, okay? You had to send a signal out your motor neurons to your arms. And you had to make it work because you didn't want, dang it. Okay? No, all of you, it wasn't like any of you like, I'm going to work on that. I'm sure I'll be able to do that. No, you're like, okay. Okay? Think about it right now. Clap once. Touch your nose. Clap twice. Touch your ear. Do you see how fast that happened? Okay? Now that's physical changes and messages and processing. So think of this like my voice, and these like your nerves processing all of this, and then here your body is going to activate it. Okay? Like when I, not it.
Not it. Oh, baby.
It took seconds, and those 2 words made you strike your neighbor. Okay? Just like that. Okay. So, come back to me. Chemical messengers, you could say, think of it like dialing out on your phone. Receptor proteins, the ones who receive the call after it bounces off a satellite. Different cells have different combinations of receptors. Different combinations of receptors. Signal transduction pathway, a series of events or relay proteins triggered by the phone caller receptor. And then youngest bio buddy, tell them what it says under response. Okay, moving on. Okay, so that was just a little interjection, and you can see the importance of the cell membrane and this process, and we will revisit that again. Okay, so there are 3 ways to cross that membrane. If I asked you to write an essay right now, what are the 3 different ways to cross a membrane? Could you come up with those 3 different ways? Tell your bio buddy right now. 3 main different ways. Okay, come up with them.
Yeah. I didn't hear you.
Okay, come back to me. Did your bio buddy say through the phospholipid bilayer? Okay, they should have. Did they say through a protein? They should have. Did they say whole membrane like endo and exocytosis? They should have. Okay? So those are your 3 main ways. Okay? Through the phospholipid, using a channel or carrier, or whole membrane. Show me right now. What are the 3 ways? Through the phospholipid, using a channel or a carrier, or whole membrane. Got it? Those are the 3 main ways. Okay, so the first one we're going to be talking about is this one. What's this one? Through the phospholipids, okay? I know, that's too much like channel, sorry. Okay, so in order to talk about that, we already know you can't be what or what? Large or charged, okay? So first thing is we want to talk about diffusion. You know what diffusion is. You're just going from a higher concentration to a lower concentration. So on your notes, diffusion, movement of molecules from a higher concentration to a lower concentration. Yes? Oh, you want me to do a new set? Okay. Sorry, we will pick up on this in a new video. Now have a good day.